Hello everyone, my name is Maria. I'm a bioinformatician and a data scientist at Day 2. Today I will talk about how graph theory can help us know more about you from your poo. But first, let me tell you about myself. I live in Tel Aviv. I have 2 cats that hate the fact I'm home this much. And in my free time, when there are no global epidemics, I love running and traveling. So, why do I want to know what's in your poo? Well, just a hobby of mine. No, I'm kidding. Let me explain. Did you know? The human microbiome is highly personalized. Different people have radically different collections of microbes. We can find microbiome on the skin, hair, vagina, nostrils, and many other locations. But 95% of our microbiota is located in our digestive tract. We have at least 1.3 to 1 bacteria cells to human cells, and even more fungi, archaea, and virus cells in our body. We're outnumbered. A rough estimation is that we have 2 million different bacteria genes in our gut, which is 100 times more than the estimated 2,000 human genes. Knowing all this, the connection to human health should not surprise you. Although we do not fully understand how the variation between different people influence wellness, our understanding of the link between the human microbiome and disease is rapidly expanding. In metabolic disease, such as obesity and type 2 diabetes, human genetics has failed to explain its tendency. In contrast, we can classify individuals as lean or obese using the gut microbiome with over 90% accuracy. In inflammatory bowel disease patients and children at risk for type 1 diabetes, we can notice altered microbiome composition, especially loss of diversity, which means those people have less species of microbes in their gut. Other conditions like C. diff infection, cancer, and autism showed an improvement of symptoms or a better response to chemotherapy after microbiome treatment. Many other conditions have links to the microbiome. Part of them is yet to be discovered. Speaking of microbiome-based treatments, a great example is the fecal microbiota transplant. It is basically when a doctor transplants feces from a healthy donor with a golden poo into another person to restore the balance of bacteria in their gut. So, what can I, as a data scientist, do with this data? The main motivation is to build predictive machine learning models for different health conditions. Once we can predict them, hopefully we can also prevent them. Finding connections and correlation can use for diagnosis, and the ability to define a good microbiome profile can contribute to treatment, like in the example of fecal microbiota transplant. For those models, I need features. What kind of features can I extract from the microbiome? We can learn a lot by knowing how much of each organism we have in our gut. Diversity levels, as I said before, can be a good indication of several conditions. Relationship patterns between organisms are critical. We also can search for different genes that leads for different functions. Mutation existence can help us understand the smallest variation in phenotypes, and we can build clusters using microbiome profiles. Now that I convinced you why I do need this data, how can I get it? It all starts from stool sample, which gives us a snapshot of the microbiome's current state. Now, we want to discover the organisms in the sample. To do so, we need to extract their genomes. What is a genome, anyway? It's a very long string made of millions of characters of the alphabet A, T, C, G. To get these genomes, we usually use a sequencing machine. Its input is a sample, and the output is the genomes of all the organisms inside the sample. One of the problems with this technology is that the machine must break the genomes into small pieces to read them correctly. So, we get lots of mixed short strings, and we need to identify them by mapping the output to massive databases of known microbes. Now, we can get all our genomes. But wait, there is a problem. Some of those reads do not map to anything we know, and we will not be able to identify them. Why couldn't we map those reads to anything? It happens when it's hard to grow the organism in lab conditions. In other cases, it happens when the organism is novel, rare, or even really small. Sometimes a known organism going through a mutation process, and now it will be harder for us to recognize it. So what will we do? We need to build new genomes. The solution emerged from an unexpected origin. Some of you may recognize it from algorithms course as the 7 Bridges of Königsberg. Königsberg residents enjoy strolling through their city, and they wondered if every part of the city could be visited by walking across each of the 7 bridges exactly once and returning to the starting point. Euler solved this question by representing each part of the city by a node and each bridge as an edge. Euler's circle is a trail in a finite graph that visits every edge exactly once and starts and ends on the same vertex. He proved that a necessary condition for Eulerian circle existence is that all vertices in the directed connected graph have the same in degree and out degree. Nicolaas de Bruijn adapted Euler's idea and formed the de Bruijn graph for solving the superstring problem. In this problem, given a set of n strings, you need to find the smallest string that contains all the substrings in the set. In our days, we use those 2 algorithms for our problem. The genome assembly. The genomic assembly is a computational process of drawing together numerous mixed short strings and organize them together in the correct order to assemble a full genome, just like de Bruijn's problem, but with genomic strings. Let me show you how to build a de Bruijn graph for a genome. Let's imagine we have lots of short strings like this one. Now, pick a k and split your strings into overlapping k-mers. In this case, I picked k equals 3. Now we get GGG, GGC, and the rest. Now, take each trimer and split it into 2 overlapping substrings of length k minus 1. In our example, each substring contains 2 characters, GGGG, GGGC, and you got a point. Now let's draw a node for each pair and a directed edge from each left substring to its right substring. You can see here the GG node with an edge to itself. GG connects to GC, GC to CC, and the same for the rest. With perfect sequencing, this procedure always yields an Eulerian graph. You can see that in this graph, the first and the last nodes are semi-balanced, and all other nodes are balanced, meaning they have the same in degree and out degree. Now we can reconstruct our sequence using the Eulerian walk from the graph. You can try it yourself. In the real problem, you do the same procedure on millions of short strings and combine them all together using overlapping nodes to a huge Eulerian graph. But there are still some challenges using the Bruijn graph for genome assembly. Sequence errors, repetitive sections, and computational cost are few of the leading challenges that makes it very hard to reconstruct some of the genomes. For example, in this case, it is impossible to know which one is the correct path. Is it the first option, which goes through the upper loop first? Or the second one, that goes through the lower loop first? Usually, the best option is to split the graph into 2 partial genomes. You can see here another example. This graph can occur when there are repetitive areas that creates a bubble in the graph. 2 paths are possible. The one that goes up and the one that goes down. In this case, we have a hard time deciding which one is the correct one, and we will have to split it once again. Before we finish, I want to give you a little example from the Segata lab, a large research group. They did an amazing job. They collected samples from all around the world and built using genome assembly pipeline 150,000 of new genomes. Most of those organisms were never described before. What if one of them is the key to prevent the next disease or epidemic? If you want to talk more about microbiome, de Bruijn graphs, or cats, please contact me on Gmail or LinkedIn. Thank you.